friends, so back again, Professor Kukreja. Yes, so in the last video also we discussed about enzyme kinetics, so I thought we'll continue with that only. There also we discussed about Km, I thought we'll go with that only again. So maybe that becomes easier, or you can relate the things probably. So if you missed out the earlier video, go check in the earlier one. So but this does not have any relation with that video anyways. You can go ahead with this video also. So here we have the question here. Yes, so today's question is that what concentration of substrate, in bracket it says relative to that of Km. So in terms of Km, like half of Km, twice of Km, 5 times of Km. So you will get in options like. So in what would be the value of the substrate in relative to the terms of Km required to reach the initial velocity rate of the reaction of 0.5 of Vmax, right? So here we are assuming that the enzyme is following the regular Michaelis-Menten kinetics, right? The Briggs-Haldane approach, what you call, right? We know that Michaelis-Menten had a equilibrium approach, right? And the Briggs-Haldane rather, they had what you call as the steady state approach. If you want to go for the derivations of these, you can find the link in the description box where you can have a complete and a detailed derivation of this particular what you call as Michaelis-Menten equation by both equilibrium as well as the steady state approach. Anyways, so after the entire derivation there, we come up to the equation of V0 is equal to Vmax into S upon Km plus S, right? All biochemists, enzymologists and students who are actually preparing for various entrances should be well versed with this equation. So yes, let me repeat. It is V0 is equal to Vmax into S upon Km plus S, right? Here it goes. So what would be the value of what you call as substrate concentration in terms of Km, they have asked, when this velocity is 0.5 Vmax. Let me substitute here. So my velocity V0 here, it would be 0.5 Vmax. So let me replace this V0 with 0.5 Vmax. Okay. So I have 0.5 Vmax is equal to Vmax into S upon Km plus S. Okay, great. So yes, something goes here. Yes, Vmax gets cancelled out. Thank you so much. So I'll let us try to reduce the things. So we reduce the unnecessary things, the repetitive things. So Vmax got cancelled out. Let me take this Km here. So it comes to 0.5 Km plus 0.5 S. Yes, it is 0.5 into Km plus S. So it is 0.5 Km plus 0.5 S is equal to that side I have now only S, right? Vmax is gone. So taking S and S to one side, so I have 0.5 Km is equal to S minus 0.5 S. So S में से 0.5 S गया. So like if you subtract 0.5 out of it, so half is gone. So you have 0.5. So it is 0.5 of Km is equal to 0.5 of S. So again 0.5 got cancelled. Wow, things became so easy. So most of the times things seem to be complicated, but they are very easy. So here I have this Km is equal to S. So 0.5 got cancelled. So Km is equal to S, right? So substrate concentration, when it is exactly equal to Km, right? Then at that value, my velocity will be half of the Vmax, right? So, oh, it was so easy. I actually solved it out because this actually comes in the definition. We all know that 0.5 Vmax is half of Vmax, right? So if you look at the graph which we had plotted earlier in an earlier video also, where you plot the velocity and your substrate concentration, half of your Vmax, that is 0.5 Vmax, is when the substrate concentration is equal to Km. I solved it out because like you can use this same if you want 5.4 times of Vmax. Can you please tell me what would be the substrate concentration in terms of Km required to reach 0.4 of Vmax with the calculations which I just done. So please stay tuned with me, Professor Kukreja, and use these simple hacks. So for 0.5 Vmax, you do not do all these calculations. For 0.5 Vmax or half of Vmax, the substrate concentration required is equal to Km. So S is equal to Km when V0 is half of your Vmax rather. So stay tuned with me, Professor Kukreja, for more in life sciences. Thank you.